Good morning, everyone. I know there are more people trickling in, but I'd like to go ahead and get started for this great session. I'm Dr. Larry Eichenfield, hailing from about 10 minutes north of here. I work at the University of California, San Diego and Rady Children's Hospital, where I'm vice chair of the Department of Dermatology and run the pediatric dermatology group. And I also serve as president-elect of the International Eczema Council, and the IEC is hosting this session today, which should really be a great session, and I'm glad to have so many people here starting off the SID meeting. So about today's meeting, one of the things that IEC does is not just put together great sets of faculty to discuss what's state of the art, in this case of the science of atopic dermatitis, but this will also be recorded and available on the web. And we'll go through some of the other activities that were done this year, and those are also available for your perusal. So our other symposia are there as well. If you have any questions or comments about the International Eczema Council, please contact Alan Stiles at astiles@eczemacouncil.org. And if you have questions about the IEC, joining the IEC, please contact him as well. And I'll tell you a little bit about the International Eczema Council and the way it is set up to influence atopic dermatitis around the world. And it basically is an international organization with an international scope. And we have counselors who are more established, if not senior, at least those people who have been multiple years in atopic dermatitis with extensive publications and influence in the field. And we have associates who are generally more junior people who are dedicated in their work to working on atopic dermatitis. And you can see from this map that we have an international scope with both counselors and associate counselors from throughout the world, with over 33 countries and 6 regions represented. And we're actually very conscious of increasing our numbers of counselors and associates from parts of the world that may not be as represented as North America and Europe on this list of individuals. So we have, IEC has 151 counselors and associates, including some of the leading voices in atopic dermatitis. The counselors are well-established and experienced clinicians or researchers. I'll mention in a moment that we've actually expanded the criteria for those who can be associates or counselors to include PhDs and not just physicians. And many of the counselors have publications in peer-reviewed journals on some of our core interventions in atopic dermatitis and actively involved in research and translational research in AD. We have 34 associates who are early career clinicians or researchers in atopic dermatitis. And you can see the distribution of the regions that are represented, as I said, with our consciousness that we'll be expanding less represented regions as well. So we're always looking for people who are dedicating part of their professional lives or personal lives on research in atopic dermatitis, especially in regions that aren't well represented. So if you're interested, you can go online, look for criteria, and consider applying for membership. So we have recently changed the bylaws of the IEC to welcome PhD members, both at the counselor and associate level. And this is in recognition of the importance that PhDs may be making in terms of our contribution to the science and literature on atopic dermatitis. Okay. So if you're a PhD you're here and you're interested in atopic dermatitis and you've been researching, consider IEC membership and grab some of the people who are part of the session today and over the weekend to ask about it, and we can tell you more about it and how to link up with the IEC. For associates and counselors who are here for the SID meeting, we will have a session tomorrow morning at 7 to 8:30. in room 400A of this floor of the hotel. The counselors get together to do a little planning and review of the work that the IEC is doing. So what's the mission of the IEC? It's basically to improve patient care by providing research, education, and best practices in treatment of atopic dermatitis for people throughout the world. Our vision is transforming the lives of people with atopic dermatitis, and our anthem is that it's a global community of leaders in atopic dermatitis research, treatment, and prevention. So the focus, of course, is changing the lives of individuals and their families with atopic dermatitis, and it's through putting together this international group of leaders. We're patient-centered, innovative, evidence-based, globally inclusive, collaborative, and forward-looking. IEC is taking the opportunity now to thank our corporate sponsors. You see our president's level, AbbVie Pfizer, our executive council, Lillian Leo, and our directors' councils included Amgen, Dermavant, Galderma Insight, Kyowa Kirin, Sanofi, and counselors Lever, Almirall, and Arcutis. And sort of a partnership with these companies supporting the work of the IEC. So the IEC is busy, and we're busy doing position papers, busy working on materials that can be resources to both clinicians and researchers around the world. We've had 47 symposia presented, nearly 150 expert speakers, 26 countries represented by speakers in the symposia, similar to today, 136 videos on YouTube, and 80 hours of free content. So it's those of you who may be working with trainees or are trainees know that the materials that are online from the symposia done last year and this year are really an incredible way to get a download of state-of-the-art material on atopic dermatitis and associated conditions. So our 2025 symposia, first one was actually in San Diego a few months ago. It was the impact of environmental, food, and contact allergies on atopic dermatitis. That was part of the Quad AI meeting that was being held in San Diego. At the AAD, we had a sort of combo, converging pathways, atopic dermatitis and alopecia areata, which was a partnership of the IEC with other groups as well. At the World Congress of Pediatric Dermatology, which just held a few weeks ago in Buenos Aires, we had a fabulous session on practical strategies for preventing, treating, and managing atopic dermatitis in pediatric populations. All three of those sessions are available online for your perusal. And today we're having our practical applications of multiomics in atopic dermatitis. And at the EADV meeting in September, there'll be a session on AD and cutaneous T-cell lymphoma, which should be quite intriguing, a controversial topic. You can find and connect with the IEC on social media, follow, like, or connect with us, and feel free to share your posts as well. You can look at the videos on the YouTube learning session to increase your own knowledge, and feel free to let other people interested in AD know about that as well. Any general questions, you can give Alan Stiles an email at astiles@eczemacouncil.org. And I think you've seen both on the chairs and up front that if you haven't grabbed the program for today's session, this is the way we're doing it. So please scan in and take a look so you can see the details of our great faculty and directors who will be running the meeting. And so right now we will begin that meeting, and I will have our great co-chair, Sarah Brown, who runs the dermatology department at the University of Edinburgh in Scotland, and Pat Brunner, one of our counselors and phenomenal scientist who's up at Mount Sinai in New York, and they will run the program. Thank you.
So, thank you, Larry, for the introduction. I'm really delighted to be co-chairing this session with my colleague Patrick here. I hope you can see from the program it's something that we've put together in quite a thoughtful way. We have this title, Practical Applications in Multiomics, and this really feeds into the aims that we have in the IEC to transform the lives of patients with AD. We want to understand this complex condition even better, and we believe that the applications of multiomics is something that is needed both now and also in the future. So I really hope you benefit from this session as much as we've enjoyed putting it together. So if I can introduce Dr. Patrick Brunner. He obtained his MD degree from the Medical University of Vienna, Austria. He completed his residency in dermatology at the Department of Dermatology at the Medical University of Vienna under the mentorship of Dr. Georg Stingel. And afterwards, Dr. Brunner joined the Laboratory for Investigative Dermatology at the Rockefeller University in New York, obtaining a master's degree in clinical and translational sciences, and he performed research on AD under the mentorship of Dr. Emma Gutmann and James Kruger. In 2018, he returned to Vienna to build a research group focusing on cutaneous immunobiology of chronic inflammatory skin diseases and primary cutaneous lymphomas to better understand how individual immune cells orchestrate skin inflammation in benign and malignant conditions. And then in 2021, he joined the Icahn School of Medicine at Mount Sinai as Associate Professor of Dermatology and Director of the Cutaneous Lymphoma Clinic at the Kimberly and Eric J. Waldman Department of Dermatology. So thank you, Patrick.
Thank you so much, and I'm very excited to be here with Sarah, and we are very happy that you all came to this program that we put together. And I'm very excited to hear about Sarah's talk. As you already heard, she's a Wellcome Trust Senior Research Fellow and Grant Chair of Dermatology at the University of Edinburgh, Scotland. And she's also an Honorary Consultant Dermatologist in NIH Lothian with specialist clinics for adult and pediatric eczema, as well as genetic skin diseases. And we are very much looking forward to her talk.
So, yeah, I get the first talk. Here we go. So, here's my title. Genetic and omic analyses give insight into fundamental mechanisms in AD. And here's my disclosures slide. I'm lucky enough to get research funding, but no personal payments. And here's an outline of what I'm hoping to cover in the next 20-25 minutes. Firstly, why do we use multiomics to study AD? Why do I think it's important? What can we learn about the genetic effects? Genetics and genomics are kind of the first step in the pathway of multiomics. And then how do we move from finding genes or genetic variants through to important functions? What are the mechanistic insights we can gain? And I'm going to give you two examples from my own work, one quite recent, one from previously published work. And then lastly, bringing it all together, what can we hope for in the future? So first up, why use multiomics? So this is atopic dermatitis. This is the disease that I guess we all struggle with in our clinics. And when you're talking to patients and families who are living with atopic dermatitis, they might ask you, what causes AD? Why do I have AD? And you can talk about families. It often runs in families. You can talk about the home environment. You can talk about environment on a global scale and the impact that might have on AD. And you can talk about the activities that we do in our everyday life. All of these things can feed into whether a patient has AD or not. And then on a scientific level, we can attempt to understand the disease at this multiomic molecular perspective. So DNA, RNA, proteins, metabolites, and lipids, we're getting more knowledge about how these might feed into causing this inflammatory skin disease. So really, the fact that AD is so complex and multifaceted is why we need to use everything in our portfolio to understand it. It really is one of the most complex human diseases arising from both genetic and environmental contributions. So unless we use this full gamut of molecular assessments combined with the clinical and epidemiological work that we do, we're never going to have a chance of understanding such a complex, multifaceted disorder. So I said I'd start with genetic effects, really because that's my area of expertise, but also because DNA is the first step in this pathway of multiomics. And Lavinia Paternoster's going to talk later in the session about how DNA might be used to leverage that start in the pathway as a potentially causative inference, but really getting to the nitty-gritty of why do we even believe that genetics is important, going back to first principles. We know that atopic dermatitis and other atopic diseases cluster within families. And the fact that it clusters within families is really what emphasizes the heritability, the importance of your genetic makeup as a predisposition to this disease. It's not simply an environmental disease that can happen to anybody. It's a disorder that arises on the basis of genetic predisposition. And we see this most clearly in twin studies. So monozygotic twins, identical twins, have a much higher concordance, around 70-80% concordant monozygotic twins, compared to 20% concordance in dizygotic twin pairs, telling us that genetics plays a major role. As I've said, there are also environmental effects, and the environmental effects that we choose to study are based on observational epidemiology. And I think if I ask this audience, or if I ask my patients, what environmental factors affect your eczema, what do you think is important, you'd get many, many different answers. And I've put some little pictures here to help us in our thinking. But it's a very broad spectrum of different types of environmental effects, focusing early in life but progressing throughout life, that can interact with the genetic predisposition to either cause or protect against atopic dermatitis. So this complexity gives many challenges. It is, as a researcher, as a clinician, it is challenging to try and make sense of this multifaceted disorder. But the complexity and the fact that we work on the most wonderful organ to do research on, human skin, this gives us opportunities as well as challenges. So what about the opportunities in skin research? A major opportunity that we have is that we can gather large populations. Skin is visible externally, so recruiting people is relatively feasible, and we can study both patients with the condition and the general population without the condition. And when you're studying genetics, you really do need very large populations, so we're privileged in our area of specialty to be able to do that. Another brilliant advantage of skin research is that we can visualize both common and rare disorders. So atopic dermatitis is very common, but there are other genetic mimics that are rarer. This is an image from one of Ellie Sprecker's reports of SAM syndrome, the severe skin inflammation, allergies, and metabolic wasting caused by mutations in desmoglein or desmoplakin that produces an atopic dermatitis-like skin inflammation. It's very rare, but it's given us other mechanistic clues. So skin's something that we can see, it's something that we can immediately understand and gather together large populations of the common disorders, as well as immediately see and potentially gather the rare disorders that give us unique insights. Another really cool thing about skin is that we can visualize genetic variation through mosaicism. So increasingly in genetics, we're understanding that the whole human being is a mosaic. Almost all our tissues are mosaic, by which I mean cells within the same tissue have a different genetic makeup. But in circulating tissues, for example, the blood, it's harder to define this. In skin, it's visible. We can see the genetic mosaicism, for example, in these Blaschko-Linear inflammatory disorders. And lastly, the powerhouse of my lab is this ability to grow skin organoid models. So we can access skin as a tissue, we can extract the cell types of interest, and we can reform them into organoids that really do look like the tissue of origin. And this gives us tremendous opportunities to investigate in detail the true mechanistic effects of genetic perturbations. And importantly, we can study both healthy skin in the organoid as well as diseased skin, and we can match it side by side with genetic perturbations that are specific to the disease of interest compared to an organoid produced from the same genetic background. So it gives us real insight into what is normal physiology and what is pathophysiology. And this allows us to address major questions. So coming back to why we are in the IEC, we're focusing on improving the lives of patients with atopic dermatitis. So we really want to address the questions that we get from clinic. And these are, these are some of the questions that I get in my clinic. Maybe you guys do as well. If you're MDs or if you're working in the lab, this is what motivates my lab. Patients saying, why do I have eczema? That's what we call it in Scotland, eczema. Why do I have eczema or AD? Is it caused by my cat? It's a difficult question in clinic when it's a beloved family pet. Is it caused by what I'm eating? Is it caused by the washing powder? These are questions that I get quite regularly. And then a challenging question here, why don't you have better treatments? The treatment for atopic dermatitis has been revolutionised in the last 5 to 10 years, but still it doesn't help everybody. And it's a good question, why can't we do better? And my talk today, really, at the end of this talk, I hope you'll be convinced, as I am, that genetics and multiomics can help us address these important and challenging questions from the clinic. So first question, why do I have eczema? When I was at medical school, I was taught that eczema is a result of systemic inflammation that appears on the skin. So it's a systemic circulating atopic inflammation that manifests on the skin as a tissue. But then, of course, in 2006, there was this breakthrough discovery led by Alan Irvine and Erwin McLean that loss of function mutations in the skin barrier protein filaggrin very strongly predisposed to atopic dermatitis, as well as causing the monogenic skin disease ichthyosis vulgaris. And this changed our thinking to emphasize that, at least in a subset of patients, the initial start of the disease is a skin barrier defect that then secondarily leads to the skin inflammation, and this can lead to systemic inflammation. So it doesn't start internally, it starts in the skin barrier and becomes systemic inflammation. But in reality, of course, as in every progressive scientific discovery, it's not one or the other, it's both, isn't it? So some patients clearly do start with systemic inflammation that presents on the skin, and some patients start with skin inflammation that then can progress to other atopic diseases and systemic inflammation. So it's a long answer to a patient, why do I have eczema? But I do explain to my patients that it's partly because your skin is leaky, the skin is inflamed, and that's what can give you more allergies. So clearly it's not just filaggrin. This changed our understanding. It was a paradigm shift, but it's very clear that filaggrin loss of function mutations don't explain all of eczema. So in this context, we need genome-wide association studies. And again, Lavinia Paternoster later in our program is going to talk in detail about GWAS. But just to highlight, this is the output of a genome-wide association study. It's a Manhattan plot. And the skyscrapers on the Manhattan skyline represent regions of DNA that are strongly associated with that disease. And you see the biggest skyscraper includes the gene filaggrin. But in total now we have 91 loci or regions of DNA that are associated with atopic dermatitis. And of these, where we can attribute a function, many are related to skin barrier function and or inflammation within the skin, within the systemic circulation. But we really need to understand the mechanisms better, because out of those 91 loci, it is only a very small minority where we have actually mechanistic understanding from the DNA variant through to the method or the pathophysiology of abnormality, either in inflammation or skin barrier. So this comes on to the important question of how do we move from genes to function? And when I say moving from genes to function, I'm even jumping over a very challenging step, which is moving from a region of DNA to a gene. Those regions of DNA on the Manhattan plot don't always assign easily to a gene itself. So we'll just hop over that. Lavinia will talk about that challenge. But if we've got a candidate gene, how then do we understand its function? And this is where the skin organoid, as I've said, becomes very powerful. So this is an example of an organoid model cultured in my lab. I focus on keratinocyte mechanisms in atopic dermatitis on the basis that the keratinocytes are the predominant cell type in the epidermis. So focusing on the keratinocytes, we have the opportunities to switch off a single gene of interest. And as I mentioned earlier, the importance is the isogenic background. So in a skin organoid, if we make organoids from normal skin samples from human beings without atopic dermatitis, then they have an essentially normal genetic background. It means we can switch off one candidate gene and compare it with the normal tissue. The advantage of doing that, rather than getting skin samples from patients with eczema, is if they already have eczema, by definition, they have genetic and epigenetic abnormalities within their keratinocytes, that it's immediately a very complex picture. So using normal skin cells, switching off a single gene of interest, we can compare directly with the isogenic background to do detailed functional and molecular assessments. So I'm going to give you just two examples of how we've implemented this sort of analytical pathway in my lab. The first example is taking one of these GWAS loci. I mentioned it can be very challenging. You see a nice peak on the GWAS Manhattan plot, and you think, great, it's going to tell us something really clear about the genetics of atopic dermatitis. But then when you look at it in detail, this peak here on chromosome 11 is a set of variants within the DNA that are actually in a gene desert. So by gene desert, I mean a very large intergenic region of dark genome. So it's a region of DNA and we don't really know what it does. It's not a coding region. So when you're trying to assess what does this region of non-coding DNA do, you immediately look at the nearest genes. So the two nearest genes are named here. MZ is a transcriptional regulator that binds to the breast cancer gene, BRCA2. And then the second nearest gene is LRRC32, which is a transmembrane receptor protein on activated Tregs. So how do we start to assess from the gene desert which are likely to be the important, which is likely to be the most important gene? And here we can use publicly available data. So the green dots in the middle of that plot, the green little lines, are what we call the credible set. The credible set is the set of single nucleotide variants that have been highlighted from GWAS. So any of those green dots could be the actual region of DNA that is predisposing to the disease. We don't know which one it is because they're all inherited closely together. So then how do we begin to see if it's important? Well, the next, let's see if I've got a pointer here. I do, fantastic. So here we have this little pink area here and this larger pink area here. So it might look not very important to you, but it's exciting for us in that this is demonstrating histone marks within normal human keratinocytes that demonstrate that the DNA is active. So we've got a credible set of new DNA variants, and we've got some evidence that they're actually important in keratinocytes. So it gives confidence that we're not wasting our time looking at this region in the cell type of interest that we can access. And then the other way we can look at it is regulatory features. Again, this is publicly available data. And if you look at the regulatory features, you start to be able to map from a region of DNA to the genes that might be active. And unsurprisingly, our credible set mapped to actually both genes. But it maps slightly more strongly to MZ. You can see MZ's more highly active within keratinocytes than LRRC32, this gene here. So we took the approach of studying MZ and we switched off MZ by siRNA knockdown in primary keratinocytes. And sure enough, we saw a phenotypic change within the organoid. So when you're making organoids from primary cells, it's very important to replicate. It takes quite a lot of time to make organoids, but still, it's no good doing it in one donor. You have to do it in multiple because skin is such a variable organ. And these are just 3 examples. We did it in total in 12 individuals. And we were able to recapitulate and reproducibly demonstrate that when you knock down MZ, you get an increase in thickness of both the epidermis and the stratum corneum. And then importantly in the organoid, we don't just have to look at the histology, we can also do a functional assessment. So this is one example of our functional assessment, putting lucifer yellow dye on the surface and seeing whether it penetrates. And here, this is the time course for the organoid maturation. So at 3 days in normal skin, the dye penetrates straight through from the stratum corneum to the dermis. At day 5, it's still penetrating, and by day 7, we have a fully mature barrier, so the dye no longer penetrates. And then you contrast that with MZ knockdown, and here the dye is excluded from both the epidermis and the dermis by day 5. So you're getting an accelerated as well as an enhanced barrier formation when you knock down this gene. So it seems like it has the potential to protect from eczema. And then importantly, coming back to genuine samples, we stained for MZ in human AD samples, and it's this sort of brownish immunostain. So when you compare normal with eczema skin biopsies, you see there's more MZ in the nuclei, and the nuclear MZ staining extends throughout the epidermis. So trying to bring in now the multiomics, it's all very well saying we've got a DNA regulator protein in nuclei of keratinocytes that seems to be more highly expressed in eczema biopsies, but what on earth does that mean for mechanism? And here, detailed proteomics comes in, and again, we go back to the organoid model, because when you're doing very detailed multiomics, the advantage of an organoid is that you can take away all the other complexity of inter-individual variation, of environmental effects. These are artificial skin samples cultured within a very controlled laboratory environment, so we can look in detail at what exactly this one gene is doing. So when we knock down MZ, we get a massive increase in gene expression of multiple pathways that are all relevant for inhibiting the disease states of dermatitis and hyperkeratosis. And interestingly, within these pathways that are upregulated when we knock down MZ, we see multiple Mendelian skin disease genes. And I've circled them there, but these are all genes that cause skin barrier dysfunction. So putting it all together, it seems that knocking down MZ is a potential therapeutic target that could protect against these multiple genetic skin diseases as well as atopic dermatitis. And that's a therapeutic target that we're working on. So the second example relates to another clinical question. So when patients ask, is my eczema caused by my cat? What they're actually asking is, tell me about gene-environment interaction in atopic dermatitis. And this is a paper that we've put out on medRxiv just recently. But basically, we took the 24 top single nucleotide variants from the most up-to-date GWAS, and we tested for interaction between a selection of environmental factors, 10 environmental factors listed here, and these environmental factors were chosen out of the many that we could have looked at. Our choices were based on what the patients thought was important. I worked with a patient support group called Eczema Outreach Support, and they were very keen to focus on environmental factors that a family could actually do something about. We also consulted with our colleagues within the UK, and everybody has their favourite environmental factor. And then the final determinant was whether the data were available. Obviously, it's good to have ideals about what environmental factors you want to study, but at the end of the day, if the data aren't available in large enough populations, then it's just not possible. So what did we do? We collected together a large consortium of collaborators from this all these different countries, brought together largely through the EAGLE consortium to bring a total of 25,000 individual data points. And then we replicated in an even bigger analysis, nearly 200,000. And then we meta-analysed these two data sets. And humblingly, out of all this massive work that took about 4 years in total, we only found one SNP environmental interaction that I've shown you here, and it is between dog exposure in early life and this variant that is an intergenic variant, but it's near to the interleukin-7 receptor. And just to emphasize, even with these very big sample sizes, we were still only achieving nominal significance, so a p-value of less than 0.05. So you could argue that it's a very questionable finding. It was replicated in both the replication and the meta-analysis, but it remains only nominally significant. So then, what do we do next? There's no more data in the world that we're able to get our hands on, so we needed a rather different approach. So coming back to the lab work, here we have the opportunity to directly expose keratinocytes to the dog allergen to try and ask the question, is interleukin-7 receptor and dog allergen exposure relevant at all? And here, using a very simplistic exposure of dog allergen provided by a skin prick test reagent that we would use in clinic. We expose the keratinocytes, primary human keratinocytes in culture to the dog allergen, and we look at gene expression by qPCR. And you can see that some of the classic atopic cytokines are downregulated when you expose keratinocytes to dog, and many others are upregulated. So it's possible that this downregulation of TSLP and interleukin-33 might contribute in part to the protective effect that we see with dog exposure. But then what about the gene-environment interaction? So here we use genotyped keratinocytes stratified for this variant that is near to interleukin-7 receptor, and we showed that keratinocytes with a TT genotype had a bigger response than those with a CC genotype when you stimulated them with dog. So this is the initial in vitro evidence that this genetic variant may explain the fact that dog exposure modulates risk. So just to summarize the main findings, we've known from epidemiological studies, and we demonstrated this in our gene-environment interaction, that having older siblings can be protective, but it can also be risky depending on your genotype. We've shown that dog exposure in early life may be protective, and it's intriguing, it has a very similar effect to the older sibling. It's the same genetic variant, it's the same direction of effect. So you can have a dog instead of your big brother, and it can protect you against atopic dermatitis. But even larger studies are needed. You might notice I haven't asked the question, is my cat causing my eczema? Because in our studies, we didn't replicate the previously reported evidence for cat exposure causing atopic dermatitis. So for cat lovers and for dog lovers, there's potentially reassuring news. But that's not to say on an individual level, specific cat allergy doesn't exacerbate skin disease in an atopic individual. We know that's the case. This is looking at gene-environment interaction on a population level. And as I've said, even larger studies are needed and further in vitro testing to follow up on this mechanism. So finally, to wrap up, looking to the future, treating AD better really is our future. And the advantage of this massively complex disease is that there are lots of therapeutic targets. And to the clinicians in the room, these are probably quite familiar. The ones that have already been very effectively targeted, we're really privileged to use these drugs now for our patients. They have changed lives. We're not currently effectively targeting the microbiome. Work is ongoing in that area, but it's very challenging. Similarly, there are treatments in the pipeline that do seem to improve skin barrier, but nothing very specifically targeting skin barrier. And that's a promising opportunity for the future, both to prevent atopic dermatitis and to protect from flare-ups. So lastly, just to acknowledge, as you can see, I'm inspired by patients and I'm very grateful to them for sharing their stories. Also very grateful for the skin samples that we obtained from our colleagues in the breast surgery unit and the hospitals in Edinburgh, and my brilliant lab team who've carried out the large part of this work, other collaborators more widely, and my funding bodies. Thank you very much for listening. Happy to take any questions either now or at the end.
Dr. Brown, that was brilliant. Thank you. Tough area to cover, and you did it so well. So we're in this era now, I think, where we're thinking about therapies might be disease-modifying. And I think you and I both agree that the barrier problem may be genetic or acquired. So is it an opportunity for geneticists to look at the people who can't be disease-modified by inflammatory approaches, anti-inflammatory approaches, to enrich your population for those driven more by genetic problems?
Sure, yeah. So we definitely see those in our clinics, don't we? The patients that just fail to respond to even really fantastic, very potent switch off of the atopic cytokines. And some of those will be like that because they've had a lifetime of atopic dermatitis, so their skin barrier has been shot away for all those years, partly through the inflammation and partly through our treatments. So the topical steroids that we use, we know improve inflammation but also impair skin barrier. But sure, there's a subset of patients that have, just that intractable genetic skin barrier impairment, and those are the ones that I think are likely clinically to need either primary treatment for the skin barrier or secondary treatment in combination. So you switch off the inflammatory response with the biologics that we have, and then whatever treatments we have in the future to improve skin barrier function, we can bring those in as well. But I guess I thought your question was going to go along the lines of whether we can intervene very early in life to try and prevent this horrible lifetime of suffering that we see with atopic dermatitis and the other atopic disorders. And that is somewhere that I believe genetics can help, because what you need to do is know at the beginning of life, is this patient going to follow that trajectory of severe lifelong disease, or are they going to be a patient where they have mild transient atopic dermatitis? And it's the patients that are destined genetically to have a trajectory of severe lifelong disease that we might want to step in proactively with more aggressive anti-inflammatory treatment and more aggressive skin barrier repair, skin barrier maintenance. In the interest of time, keeping to our program, I'll hand over to our next speaker, which is our co-chair, Patrick. Over to you.
Thank you so much, and I'm excited to talk about AD and its mimickers. These are my disclosures. So you're all very well aware of the fact that atopic dermatitis is quite a heterogeneous disease. Thus, early in life, some patients outgrow the disease, some don't. We don't really know why. And depending on the group investigated, there can be quite a bit of heterogeneity as to the cytokine profile that you find in the skin. But overall, what we know and what has been demonstrated so far, atopic dermatitis is a type 2 driven disease. Type 2 activation or type 2 Th2, TC2 hyperactivation is present in all populations investigated, and that really seems to be the backbone of the condition. But talking about atopic dermatitis, we also need to talk about rashes that we often cannot really diagnose. There are those patients that show an inflammatory rash that doesn't really fit to atopic dermatitis or that might mimic atopic dermatitis and later on turns out to be something quite different. And many of those, or some of those rashes that we see cannot even be properly categorized in certain instances. And I think tissue profiling now allows us to better look into those patients. And I think molecular profiling, we're not really there yet, but I think molecular profiling will allow us to better understand atopic dermatitis and other rashes that mimic atopic dermatitis that might be a different condition. And as we see with current targeted treatment, some patients respond very well to targeted treatment, some others don't, and the ones that don't might just have a different condition. And in this regard, I think single-cell RNA sequencing has really revolutionized the field. It might not be a method for routine clinical use, but for sure it is a method that now allows us to look at skin biopsies at unprecedented depth and will likely allow us to identify biomarkers that we can then specifically use for better diagnostic purposes. There was this paper in 2022 that really, I think, kicked off a lot of research in the field where single-cell RNA sequencing was used in order to profile healthy control skin versus atopic dermatitis, psoriasis, and other rashes. This is a typical single-cell RNA-seq plot where each dot represents one cell. And the good thing about single-cell RNA sequencing is you can look at the transcriptome or the genetic expression within each cell, and with bioinformatics algorithms, you can cluster those cells together and compare different patient populations, different biopsies at a very nuanced level. And importantly, you see which cell produces what kind of cytokine. And then you can apply certain algorithms, you can apply certain bioinformatic processes in order to cluster certain patients, certain biopsies in the vicinity to other conditions. As you can see here, there is a group of patients that have an unclear rash here in yellow, and it was clustered next to psoriasis and atopic dermatitis. And you can see some of these unclear rashes clustered within psoriasis, the others clustered within AD. And I think with such a method, we will be able to profile skin better in the future and particularly hopefully find better therapeutic targets in order to tailor treatments to individual patients and not so much a try and error approach that we have right now. When we talk about atopic dermatitis and its mimickers, I think we need to talk about cutaneous T-cell lymphomas, we need to talk about mycosis fungoides, and as you can see in this example of a photographic atlas that was introduced about 100 years ago by George Henry Fox. He did the first photographic atlas in dermatology. He writes that mycosis fungoides or a patient that suffered from mycosis fungoides had, for the most part of his year, typical eczema. So even back then, this has been, it's a clinical challenge for all of us that mycosis fungoides cutaneous T-cell lymphomas can mimic atopic dermatitis, can mimic other chronic inflammatory skin diseases such as psoriasis. And particularly early stage mycosis fungoides is often very difficult to differentiate from atopic dermatitis. Why is that? Because not only clinically it can be challenging, also molecularly, particularly when the disease progresses, there's a strong increase in type 2 Th2 cytokine profiles within these patients, and especially then in advanced stage disease on a molecular level, it's often difficult to differentiate those two conditions. When we talk about advanced stage mycosis fungoides, advanced stage CTCL, when we have patients that have growing tumors, of course, no one's gonna mix that up with atopic dermatitis. But there are those patients that come with erythroderma that might not have Sézary cells in the circulation, but the biopsy doesn't really show much. So erythroderma can be a big challenge to diagnose or properly make a diagnosis, particularly because there's also one patient population that shows erythroderma, patients that are red from head to toe, where you cannot really make a diagnosis. So that patient suffers from month-long erythroderma. There was no clonality in the peripheral blood. The biopsies, skin biopsies, didn't show any specific features suggestive of cutaneous lymphomas, was just spongiodic dermatitis, and dupilumab didn't really work. And there are those patients with erythroderma where we cannot really make a diagnosis and where you end up calling them patients with chronic idiopathic erythroderma. So we were interested in those patients and we did some tissue profiling, we did single-cell RNA sequencing with those patients. And here on top I show you the T-cell cluster or the T-cell population of those patients with chronic idiopathic erythroderma, and we compare it with atopic dermatitis, healthy controls, and erythrodermic CTCL, a mixture of, in this case, erythrodermic MF or Sézary syndrome. And we performed single-cell RNA sequencing, and the good thing about this method is that you can also do T-cell receptor sequencing. You get information of the specific T-cell receptor for every cell, and then you can see, is there an expanded clone or is there no expanded clone? And in this case, we colored the polyclonal infiltrate in those patients in green, and the single top expanded clone was colored here in red in erythrodermic cutaneous lymphoma, and you see massive expansion of a top clone, so most of it is red, which means that's a single top clone. In healthy controls, the top clone, or so-called top clone, is just a stochastic clone that is there at higher levels, and it evenly distributes across this cluster. I'm not sure you can see this, but overall it's a polyclonal infiltrate with just a few dots of a top expanded clone. In atopic dermatitis and in chronic idiopathic erythroderma, there was a bit of an oligoclonal expansion in those patients, but it was far away from the massively expanded clone that we see in lymphomas. And in the middle panel, you see the amount of the top expanded clone, pretty much the same in atopic dermatitis and chronic idiopathic erythroderma as opposed to the lymphoma patient. And then, single-cell RNA sequencing allows you to profile those patients, and as expected, atopic dermatitis showed a massive expansion, or showed a lot of interleukin-13, whereas this chronic idiopathic erythroderma patients rather showed more of a type 1 immune skewing that was completely different from both the lymphoma patient as well as the atopic dermatitis patients, was also very distinct from psoriasis, which I do not show you here. So we believe that this group of chronic idiopathic erythroderma patients indeed is a separate population. And we found, to make a long story short, we found a CD8 positive population that expresses NKG2D both in the blood and in the skin, that showed some oligoclonal expansion, particularly in the blood, that was far away from the lymphoma, but still some expansion. We therefore believe that these patients form a separate entity, and when you stain for this marker in the skin, you see that there's quite a substantial number of these cells in those patients, as opposed to all the other diagnoses that we investigated. So I think that kind of chronic idiopathic erythroderma is a separate nosological entity. So this was as for advanced stage lymphoma. What about early stage lymphoma? So there's often it's often very difficult to make the diagnosis of early stage mycosis fungoides, and there are those diagnosis terms, floating around that have been introduced 100, 120 years ago, like parapsoriasis, and we do not really have a good definition what it really is. Some colleagues will say parapsoriasis should be abandoned, it doesn't mean anything, it's just early lymphoma. Others say it might be a useful term for patients that show a rash that we cannot categorize, but where we also cannot really make a diagnosis of lymphoma. So this is why we did some tissue profiling on these patients. So here in this case, we took bona fide atopic dermatitis patients, early onset disease, atopic sensitization, typical history, and compared it to two patient populations that walked into our lymphoma clinic that showed two phenotypes: a rash with small patches and a rash with large patches. And just ignoring everything else, we categorized those patients into small patch and large patch dermatitis. Why did we do that? Because small patch and large patch parapsoriasis has been categorized in the past, and there is some risk of developing overt mycosis fungoides, particularly in patients with large plaque parapsoriasis that was higher. Historic data show that the risk of developing lymphoma is particularly high in large plaque parapsoriasis than in small plaque parapsoriasis. So when we just compare those patients in the lymphoma clinic with those patch-like lesions and categorize them in small and large plaque, consistent with what we know from historic data, when you look at large plaque dermatitis lesions, many of them have an expanded clone as shown in the bottom here. So the dark red shows the top expanded clone, the light red shows the polyclonal infiltrate, the percentage within a sample. So most of the large plaque patients showed an expanded clone. The small patch dermatitis or small patch parapsoriasis patients, only one had an expanded clone. So just clinically, I think those patients can be differentiated. It might be that large plaque parapsoriasis is just identical to early stage mycosis fungoides, whereas small plaque parapsoriasis seems to be something different, because I believe that in order to really make a diagnosis of lymphoma, you need to have an expanded clone. I think this is the strongest evidence that we have to make a lymphoma diagnosis. And also, to make a long story short, we of course looked at those patients with more small plaque parapsoriasis that we here term just parapsoriasis, as opposed to patients that had an expanded clone. And within all the cell populations that we investigated in the skin, we found a tiny little population that was very unique to small plaque parapsoriasis or polyclonal parapsoriasis. That was an innate lymphoid cell population that co-expressed a neuropeptide Y and CRISP3, and we were able to corroborate those populations, CD3 negative, NPY positive population in the skin, and this was a unique ILC population that only was present in those polyclonal small plaque parapsoriasis patients, and that's why I also think that this condition, particularly when it shows as sort of fingerprint dermatitis, is a separate entity that has nothing to do with early stage MF and also has nothing to do with eczema, psoriasis, and so on. And maybe this NPY will help as a biomarker in the future to make a more solid diagnosis for those patients. Prurigo nodularis, of course, is not necessarily a mimic of AD because, of course, it looks very distinctively, but of course it can happen on the background of atopy. It can respond to the same treatments that we do in atopic dermatitis, but pathophysiologically, most likely there's something else going on. Most of those nodules are, of course, induced by scratching, by some mechanistic stimulus, but we also are interested in better profiling those patients because, again, with these methods now, it's possible to lay out all the T cells in those three conditions, chronic nodular prurigo, AD, and healthy control, and we can nicely compare cytokine expression profiles in those patients. And as you can see here, interleukin-13 is highly upregulated in atopic dermatitis, was also somewhat present in chronic nodular prurigo, but was present at higher levels than in healthy control. So there seems to be also, or we also found the type 2 bias, which is of course consistent with other published data and with the fact that type 2 blocking agents can work in this condition. But there was one thing that we found quite interesting. When we looked at the T cell receptor positive cells, so the repertoire of T cell receptor positive cells, and compared it to chronic nodular prurigo and atopic dermatitis, we saw, and I briefly mentioned it already, there is a bit of an oligoclonal expansion in atopic dermatitis of T cells that was higher, not a lot, but consistently higher across all patients than in healthy controls or in chronic nodular prurigo. So when you just display the top 10 clones in atopic dermatitis, in almost all patients there was this oligoclonal expansion in AD that we did not find in those other conditions. You might argue this is maybe a sampling error, maybe those T cells just get out more easily from the skin in AD, so maybe that's just an isolation artifact. But I believe that it's not an isolation artifact because when we look at the clustering behavior in the middle top of those different conditions and those top clones, we see that the atopic dermatitis, oligoclonal expanded cells, they cluster very neatly and very consistently in the center of this cluster. So I don't believe that this is just by luck or by chance. It seems to be a specific expansion within specific clusters that might be T cell receptor mediated, that might be due to allergic sensitization via the environment. And this is something we did not find in other conditions, including chronic nodular prurigo or healthy controls. And those oligoclonal expanded samples showed interleukin-13 expression consistent with the fact that we find that, of course, in atopic dermatitis. With all these multiomics and tissue profiling, you can then, of course, sort of digest all the information that you have in certain pathway analysis. And when you compare, for example, atopic dermatitis with chronic nodular prurigo, you see that you get a lot of T cell receptor regulation in atopic dermatitis, whereas chronic nodular prurigo showed more of a fibrotic pattern, which is, of course, perfectly in line with what we see clinically. And one interesting thing that we saw, that the itch mediator neuromedin B was elevated in chronic nodular prurigo fibroblasts, and we found its receptor on nerve endings. So there might be a crosstalk between fibroblasts and nerve endings, or mediators that fibroblasts produce that can induce and perpetuate itch, something that we did not find in atopic dermatitis. And we also found that certain itch mediators were produced by endothelial cells among all cells. So also those cells potentially, or one might speculate that also those sort of stromal cells are part of an itch-scratch cycle in chronic nodular prurigo patients. To better understand what's going on in atopic dermatitis, we were not only interested in profiling the untreated condition, we also wanted to see what's actually happening when we treat patients, what's happening when we treat those patients with dupilumab, for example. Because dupilumab, of course, is an efficacious drug that works in a good portion of patients, but once you stop the medication, the disease sooner or later comes back, so there must be some sort of tissue memory that is there, and maybe this tissue memory is very specific for atopic dermatitis and might not be the same in other diseases, in mimickers of AD, and of course, when you want to go for a cure of atopic dermatitis, you want to eliminate the tissue-resident memory cells, the tissue memory on long-term in order to potentially bring those patients into long-term remission. So when we investigated atopic dermatitis patients that were successfully treated with dupilumab for 1 year, we found essentially almost all inflammatory cells to disappear except for one specific Th2 population that was still present even after 1 year of dupilumab treatment. That was a type 2 Th2 IL-13 producing cell that had a certain repertoire of alarmin receptors on its surface. So alarmins are mediators that are being produced by the epidermis. You scratch your skin, alarmins are being produced. And those alarmins, of course, need a receptor in order to work. And interestingly enough, those alarmin receptors were highly expressed on those Th2 cells, which have previously been termed Th2A cells. So those memory cells that sit in the skin that you cannot get rid of might be able to easily wake up once the treatment is discontinued and might mediate the flare of the disease. These cells have been previously mentioned in the context of food allergy. So when you treat patients with a specific immunotherapy for peanut allergy, those cells disappear. So there might be, this is not approved, but circumstantial evidence that those cells might be sort of important for a disease memory, maybe in food allergy, maybe also in atopic dermatitis. But why am I showing you all of that? So this was a way to detect this cell population, Th2A population. When we look at immunohistochemistry, 2 good markers were CD3 and CRTH2. And all the diseases that we investigated, this CRTH2 CD3 positive cell seems to be really present only in atopic dermatitis. We did not find these cells elevated in parapsoriasis patients or small plaque parapsoriasis patients. We also didn't find it in early stage mycosis fungoides patient, also not in healthy control. So I think so far the populations that we investigated, these Th2A cells seem to be pretty consistently upregulated in bona fide atopic dermatitis and not in other conditions. Talking about mimickers of atopic dermatitis, when you treat patients, sometimes you get paradoxical effects, like paradoxical psoriasis with TNF-alpha blockers. You also get dupilumab-associated head neck dermatitis when you treat patients with dupilumab, aside from conjunctivitis and blepharitis. And sometimes it's difficult to differentiate, is it a therapeutic failure or is it something that newly appears? So when the patient tells you, I never had this kind of rash in my face, and when I drink alcohol it gets worse, you want to think about maybe this patient suffers from this type 2 blockade-associated head neck dermatitis. We did a biopsy study in these patients, and we compared this facial rash with untreated atopic dermatitis from the head neck area as a sort of comparison within the body location, and we compared it to untreated truncated and healthy controls. And talking about oligoclonal expansion, in these patients we found strongest evidence and strongest amount of oligoclonal expansion when we looked at the skin. So when we sort of quantify the top 10 T-cell receptor positive clones, we saw that this kind of oligoclonal expansion was strongest in dupilumab-associated head neck dermatitis, and those top clones strongly expressed interleukin-13, despite the fact that those were treated with an IL-4 receptor alpha blocker. But one interesting thing was those clones that appeared in your face, that are appearing in your face of this dupilumab-associated head neck dermatitis, those oligoclones strongly upregulated interleukin-22 and AHR. And express interleukin-13 at the same time. So from these data, we conclude that IL-22 plays a role in head neck dermatitis, maybe in connection with malassezia colonization, but those cells co-expressed IL-13 and IL-22, so they are a mixture, blended phenotype of Th22 and Th2 cells, and there might be even some some T cell plasticity, where a type 2 cell becomes a type 22 cell. So all these buckets, Th1, Th2, Th17, Th22, in vivo, they're much more fluid than those artificial categories that we are looking at. And time will tell whether IL-22 blockade might help prevent this kind of side effect. We also found these cells with confirmatory immunohistochemistry, stainings, and most of those IL-22 positive oligoclones that also expressed IL-13 were actually not CD4, but CD8 positive cells. So with this slide, I want to conclude. I want to thank my group and my collaborators and my funders, and thanks so much for the opportunity to talk today. Thanks. Any questions so far?
Patrick, really nice talk. Lisa Beck, Rochester. In your oligoclonal analysis early in the talk, comparing AD to other AD mimickers, thought it was interesting you found them primarily in CD8 cells and not in CD4. What do you think is the relative import of CD8 in the pathophysiology versus CD4?
Yeah, no, thanks for pointing this out. So that's what we see quite consistently in those inflammatory conditions. And when we compare it, for example, to healthy control skin, if we enumerate the top expanded clone, which of course is not really, it's top but not expanded, those are usually CD4, CD8 mixture, CD4, but in these inflammatory conditions, in atopic dermatitis, but also in this chronic idiopathic erythroderma, not in every patient, but quite consistently, most of them are CD8 positives, particularly in the blood of these erythroderma patients. So we don't know what it means, but that also sort of gives us confidence that this might be a consistent, true pathophysiologically relevant phenotype and not just the sampling error that we do. Yeah. But interestingly enough, the CD8 population, of course, is also neglected. We always talk about Th2, but in fact, it's type 2 that encompasses many more cells, ILCs, CD8 positives, CD4 positives, and so on. Absolutely, yeah.
Luis Santa Maria from Barcelona. I have two questions, and this is related to the translational relevance of some results. We always focus on resident memory T cells, but there are ex-resident memory T cells circulating. This is the first question. What's the relevance of ex-resident memory T cells in atopic dermatitis? And the second question is, Th2A depends on alarmins, and we know that blocking alarmins in the clinic do not provide any improvement in the clinic. So what's the relevance of those cells?
You're talking about tissue-resident memory T cells and alarmin receptors. If you block them, it doesn't go away. Yeah, so there might always be, if we find a biomarker, there might always be redundancy, and just because those cells express the biomarkers, by no means does that by blocking them, does anything, like also with chemokine receptors. Tissue-resident memory T cells, I think, are important for tissue memory, but, there's a lot of, we learned a lot of new things about these cells recently. They might not just be ended up in the skin. There's some evidence that they can recirculate. So all those categories that we have for sure are oversimplifications, and time will tell. Particularly, I think, those profiling studies in real patients that have the real disease and not model systems, murine model systems that might be a little bit too artificial. So hopefully with looking and profiling humans with the actual disease will give us better insight in what's really also happening, not only in the condition, but also in the human immune system altogether.
Thank you.
Hi, I'm Yang Wang from Peking University First Hospital. And currently, there are more and more reports demonstrating that dupilumab treatment after dupilumab treatment induced mycosis fungoides in atopic dermatitis patient. So do you think it's the unmask of the underdiagnosed mycosis fungoides, or dupilumab somehow promoted the clonal expanded T cells in atopic dermatitis to mycosis fungoides? Thank you.
No, that's a very, very important topic. We actually have an IEC symposium at the EADV in Paris about exactly that topic. But, whether dupilumab causes lymphoma is very controversial. I mean, there are three options, right? It doesn't do anything. It demasks and misdiagnosed mycosis fungoides, or it really leads to malignant transformation. So when you look at all those data out, there's for sure a heavy, publication bias towards patients that have a rash, that have been treated for AD, and that then got worse. So what are those patients, right? I believe from my experience, and also when you look at the published data, there have also a recent meta-analysis, most of those patients that are treated with dupilumab, they don't get better and then are diagnosed with mycosis fungoides. Almost all of them are misdiagnoses, initial misdiagnoses when you look at the data. So so-called late onset atopic dermatitis, no atopic comorbidities, and so on. So when you break it down, most of those patients had mycosis fungoides all along, were misdiagnosed because it's often difficult to diagnose, also on a histopathologic level, then got dupilumab. I believe dupilumab doesn't work in mycosis fungoides, just doesn't do anything. And then, you either have a treatment-resistant patient or even you have the natural cause of the disease, the patient gets worse. So I think most of those patients are misdiagnosed. There's this concept that the scavenger receptor plays a role in IL-13 signaling. I'm not so sure whether that's really a true cause, but that's of course an interesting thing to investigate. But all in all, I think most of these patients are just misdiagnosed early on. And on the other hand, when you look at sort of misdiagnosed patients, you have those patients that you treat with dupilumab or any type 2 blocking agent that then get or are diagnosed mycosis fungoides, and then you say, I'm unmasking or so. Those patients that are misdiagnosed and that respond to dupilumab despite their mycosis fungoides because they might have a subset that responds to dupilumab, those patients respond, they will never be identified. We will never know about these patients because those patients will never be rebiopsied or their diagnosis will never be questioned. So I think there's a big portion of a publication bias. And as for the data that are out there, we cannot answer it 100%, but most of these patients, I think, are misdiagnosed. Also, when you compare dupilumab-treated patients with other treated cohorts, right? When you compare to methotrexate, when you compare to prednisone, there's not really an increased rate of lymphoma development in those patients. And when you look at all those dupilumab-induced lymphomas, right? It's only happening in atopic dermatitis. It's only happening when you have skin involvement, right? It doesn't happen when you give dupilumab for other conditions such as asthma without skin condition. So if you really have an agent that really triggers or causes lymphoma in a significant amount of patients, why does it only cause mycosis fungoides in internal lymphomas? And why does it never cause mycosis fungoides in patients that never had a skin condition before? So the circumstantial evidence also speaks very much for the fact that all those patients, or most of those patients, are just misdiagnosed and did not de novo develop
Thank you very much.
mycosis fungoides.
Up really quickly on that. I mean, your historical paper on MF, implied that they originally saw that it could be post an atopic dermatitis presentation. Clearly, that's true in the literature, and CTCL centers see more AD patients transitioning to CTCL than psoriasis. I wonder if your observation that you had more oligoclonal T cells in atopic derm are the reason why that's a at-risk population, even if they are absolutely classic atopics, to progress to CTCL. And what Dupi, and now we've got a case report of anti-IL-13s resulting in a CTCL, and we have JAK inhibitors now also doing that, is whether those stop some check and balance on T cell clonal proliferation, which would be a greater risk for a population that already starts with more clones.
Yes, so there's one publication that looked at dupilumab-resistant atopic dermatitis patients, and many of them were clonal to begin with. And I would speculate that those patients, that you might be able to detect the clone early on, and that might then, cause a lymphoma. But that's speculation, but I would assume that that's the case, yes. All right, thank you so much. And I'm very, very happy to call Dr. Helen Hay on stage. She's assistant professor and a fellow colleague at the Department of Dermatology at the Icahn School of Medicine at Mount Sinai. And she's the co-director of the Mount Sinai Clinique Health Skin Dermatology Center. And she's going to talk about minimally invasive sampling methods in AD. Thank you so much.
Good morning. Thank you so much to Dr. Brunner and Dr. Brown for inviting me to this wonderful symposium today. And today my talk will be on minimally invasive approaches for obtaining samples to study atopic dermatitis. Here are my non-relevant disclosures. So, as we all know, atopic dermatitis is currently undergoing an exciting translational revolution, where due to our increased mechanistic understanding of the disease, there are more treatment options available now than ever before, both on the market and currently somewhere down the pipeline, including in clinical trials. And I think a large reason that we've been able to get to this point is owing to this translational bench-to-bedside research approach, where at the bench, molecular studies, genetic studies, ones we've already heard about today already, using human tissue samples, is being used to formulate hypotheses about disease pathogenesis, which then provides the rationale for clinical trials of targeted treatment options. And through multiple cycles of this has successfully facilitated the development of novel disease-modifying specific treatments which are more available now than ever before. And in order to, integral to this strategy is the need for reliable and useful biomarkers. And to get these biomarkers, we really need to think critically about how we sample the tissue. We want biomarkers that are dysregulated in AD at baseline. We want biomarkers that respond to different types of treatments, biomarkers that can be correlated with clinical data, such as disease severity. And traditionally, biopsies have been the gold standard for skin sampling for molecular profiling. And while biopsies do provide really high-quality molecular data that has elucidated disease biomarkers and led to therapeutic advances in clinical trials, there are many limitations to biopsies. Biopsies are painful, they can bleed, they scar, they can cause dyspigmentation, they can get infected. They're not really feasible for longitudinal studies where you may need to take multiple biopsies at different time points to track therapeutic response or disease activity. You're really not able to sample cosmetically sensitive areas like the face, and they're not really practical for pediatric studies either. And ultimately, these disadvantages really limit the recruitment numbers that are needed for large-scale studies and clinical trials. So there is definitely a huge need for minimally invasive sampling alternatives. In addition, AD is also such a heterogeneous disease, so no two patients present the same. Patients also respond differently to different treatments. Not everyone's going to respond to the same treatments. So to fully capture the heterogeneity of the AD disease spectrum, you need large-scale studies that where you collect samples from a larger number of patients across the entire heterogeneity of the AD disease spectrum. So again, for all of these reasons, we need minimally invasive approaches. So I think tape strips in recent years have really emerged to fill this unmet need. And a lot of my talk now is going to be focused on tape stripping. So tape strips are adhesives that are applied consecutively to the skin to sample the outermost epidermal layers, take those cells from the stratum corneum. And tape strips have the advantage, unlike biopsies, that they're really easy to implement. They're not very user-dependent at all. Painless, no bleeding, scarring, or infection. For our studies, we typically take 16 to 20 tape strips altogether. We try to sample both lesional and non-lesional tissue, non-lesional referring to the clinically unaffected appearing tissue in normal health, in AD patients. And then we use a pressurizer device for about 5 seconds to ensure that standardized pressure is applied. We used to discard the first tape. We've kind of tried it both ways, thinking that the first tape may have environmental influences that impact the data. But we found that when we discarded the first tape, when we kept the first tape, it really didn't impact the data in any meaningful way. The results were pretty similar. You just get less material to work with. So as a result, we've stopped discarding the first tape because every little bit of material is precious to us. And then when we take 20 tapes, we usually reserve tapes number 3 and 18 for proteomic analysis. The rest we pull together for RNA analysis. And for our analyses, we typically need about 5 nanograms of RNA, 10 nanograms of protein. But as you will see, even with these very small RNA and protein quantities, we are still able to get strong, reproducible, and statistically significant results. And then with our strategy of 20 tape strips, we've seen in histology that we usually are able to sample the entire stratum corneum, sometimes getting up to the upper granular layer as well. And using RNA-seq, we found that tape strips are able to capture the disease signature of AD and psoriasis, including the molecular features that distinguish them. We also built a gene classifier showing that iNOS or NOS2 is a single gene that was able to discriminate psoriasis from AD with almost perfect 100% accuracy. We also, of course, directly wanted to compare tape strips to biopsies in the same patient, so an intra-patient comparison, so just to see how tape strips stack up against the gold standard of biopsies. So what we found is that both tape strips and biopsies really are able to capture the disease signature of AD. And then here in this word cloud, all the genes that you see are differentially expressed in that respective technique. The size of the word represents the magnitude of the fold change in the comparison of AD versus controls. And there are some differences. So biopsies, for example, were really best for capturing markers of epidermal hyperplasia, S100As, that are probably located a little bit lower in the epidermis, like the suprabasal layers, which is below the collection of tape strips. Similarly, dermal cytokines like IL-22, much better captured in biopsies. But tape strips actually outperformed biopsies in several different immune markers, including in important Th2 biomarkers, such as IL-13, CCL17, CCR4. And I think the reason for this is because even though some of these cytokines are being produced in deeper layers of skin, they appear to be diffusing into the stratum corneum, where they're probably constituting a higher concentration of the total genetic material in these tiny tape strip samples compared to in biopsies with more cell types, bigger tissues. Their concentrations are probably getting diluted. I just also want to note that IL-13, the itch cytokine, was only really captured significantly in tape strips in this analysis. And more in general, we found that several immune, itch-related, and barrier markers were actually better captured either preferentially or uniquely in tape strips compared to in biopsies. This was really striking for many markers that are known to mediate itch, IL-31, ion channels. A lot of them were only detected in tape strips. Similarly, a lot of barrier markers were distinct to tape strips, which kind of also makes sense since with tape stripping, you are focusing on the stratum corneum outer layer. So this was another study that also did an intra-patient comparison between tape strips and biopsies, and similarly to what we found, even though the RNA yield was so much lower in tape strips, 22 nanograms compared to over 4,000 in biopsies, they actually detected more genes that were differentially expressed in tape strips, where tape strips showed better detection of various immune markers, including biomarkers of Th2, while biopsies were a little bit better at some of these more structural products, such as keratins and collagens. So when designing a biomarker study, I do think this should be one of the considerations. What markers are of greatest interest and where in the skin those markers are best detected. So if you want to focus on Th2 biomarkers or perhaps markers of itch, then tape strips are a really great option. But if it's really important to capture epidermal hyperplasia or Th22 or collagen, where we are having increasing knowledge that fibroblasts play a huge role in AD, then biopsies may be important to include. So we're doing tape strips. How many tape strips do we need to take? There's a lot of variation for this across different studies. This is an interesting study from Dr. Agner's group, where they measure the protein concentration of various cytokines across the different depths, 5 different depths of the stratum corneum. And they found that the concentration was actually relatively stable across the stratum corneum, which suggests that the number of tapes you want to take probably more so depends on what analyses you want to do, and you just want to make sure you have enough material to do your analyses. So in our case, 20 tapes gives us sufficient material to do both our transcriptomic and the proteomic analyses. So as I alluded to, one area where I think tape strips fill a major void is in pediatric studies. So here we took tape strips from infants with and without AD, so less than 5 years of age, did RNA-seq and detected over 2,000 genes that were differentially expressed between infants with and without AD. Once again, built a gene classifier, which showed several genes where it was able to discriminate AD from non-AD pretty well, among which the negative immune regulator IL-34 was the best performing with an accuracy of about 94%. Tape strips were also sensitive enough to detect molecular differences in AD patients between infants, children, and adolescents, suggesting that tape strips are able to define the molecular phenotypic changes across childhood maturation of AD. So this gives us insights into the heterogeneity of AD. And since then, we've also done multiple other studies in tape strips to better define this distinct AD molecular endotypes across AD heterogeneity, including patients of different ethnicities, skin types, severity levels, and more. And you will actually hear about some of that data later on in a flash talk, so stay tuned for that. Tape strips are also really promising for defining minimally invasive biomarkers of disease severity. So across multiple studies, CCL17 has emerged as one of the most reliable biomarkers of disease severity, correlating with severity scores in multiple studies in adults and in children. In adults in particular, tape strip CCL17 levels showed strong correlations with local skin symptoms, erythema, edema, oozing, also with serum IgE levels and eosinophil counts in the blood, and also notably, serum CCL17 was also shown to correlate with tape strip CCL17 levels, as well as with disease severity scores in both children and in adults. And then here are some other biomarkers across different studies that were shown to correlate well with AD disease severity in tape strips in infants, children, and adults. So tape strips really are a promising tool for evaluating and identifying new minimally invasive biomarkers of disease severity, which has applications and implications in disease management and monitoring disease activity. We've also had great success in using tape strips to detect therapeutic and monitor therapeutic response. So in this study here, we took lesional and non-lesional tape strips from 80 patients before and after 16 weeks of dupilumab therapy and did RNA-seq. As you can see, the inflammatory tone of lesional AD before treatment, very red and inflamed as we would expect, slightly less so for non-lesional AD. But after dupilumab, we see improvement of that inflammatory tone detected in tape strips towards the non-inflammatory phenotype of healthy controls. And just like in biopsy, we saw improvement in markers of general inflammation, T cell activity, dendritic cells, Th2, with upregulation of negative immune regulators. We did something very similar using proteomic analysis as well, where we use the multiplex Olink platform that evaluates targeted protein panels So here with this platform, we evaluated over 350 proteins across 4 panels related to general neurologic and cardiovascular inflammation, where we saw that dupilumab suppressed Th2, Th17, and also interestingly proteins related to atherosclerosis and cardiovascular disease signaling, suggesting that dupilumab may be modulating the cardiovascular inflammation and risk inherent to AD patients, and tape strips are able to detect this. Also notably, no changes were observed in the Th1 pathway, which is actually exactly what we saw with biopsy, so that was very validating as well. We've also looked at tape strips before and after topicals. So in this case, after emollients, tape strips were shown to improve various inflammatory markers, including of Th2, where CCL17 and IL-8 improvements were shown to correlate most with improvements with disease severity. We also had looked at tape strips before and after topical steroids, in this case flumetazone 0.1% cream, which showed improvements in Th2, Th17, Th22, and upregulation of terminal differentiation gene products. And finally, one other study I'll reference, doing tape strips before and after topical steroids, where they identify IL-8 as the marker that correlated best with disease severity, and improvement in IL-8 correlated best with improvement in severity scores. So this here is a study from Donald Young's group, which I think is actually a very good example of using a multi-omics approach with tape stripping to study specific endotypes of AD, in this case, AD disease comorbidities. So they assess patients with AD with and without food allergies. They integrated together transcriptomics, proteomics, lipidomics, and microbiome data, and they combined it all in a network analysis where they found that abnormalities in keratin expression and filaggrin breakdown products were the features that were most associated with increased risk of AD with concurrent food allergy, as well as with increased transepidermal water loss. Another study from their group that kind of demonstrated the utility of tape strips for their predictive ability. So in this study, infants who did not yet have AD were followed from 2 months old until 2 years old. Tape strips were taken at 2 months old, and these infants were evaluated for the development of AD at 2 months old. So combining mass spec and lipidomics, they found that certain lipid abnormalities, such as decreased ceramides, increased sphingomyelins, as well as cytokine elevations in IL-13 and TSLP, were associated with an increased risk of developing AD at age 2 years old. Another study here by Dr. Tyson's group, also evaluating the predictive value of tape strips, showed that at age 2 months old, and they evaluated a pretty large cohort of over 400 infants here, but at age 2 months old, elevations in CCL17 in tape strips was associated with an increased risk of developing AD, and particularly moderate to severe AD by age 2 years old. Finally, for tape strips, I want to discuss a recent study that we did that showed some interesting results, in my opinion, where we looked at asthma patients who actually did not have comorbid atopic dermatitis or any skin manifestations for that matter. But despite this, these asthma patients, their tape strips actually showed immune abnormalities, and even more surprisingly, evidence of barrier dysregulation as well, which suggests to us that perhaps the barrier impairment detected by tape strips in these asthma patients without skin manifestations can potentially serve as a proxy for the barrier impairment in the lung in these asthma patients. In the last few minutes, I want to touch a little bit upon blood biomarkers, starting with this study here, which was an earlier study that was spearheaded by Dr. Bruner, actually, where we evaluated moderate to severe AD patients, performed serum proteomics using Olink, and really showed us for the first time that AD is not just a skin-limited disease, but there is substantial systemic inflammation, as evidenced by the immune activation in the serum profile, as well as these increased levels of markers related to cardiovascular risk, which may also be increased in atopic dermatitis. Several of these markers also correlated with disease severity score, SCORAD, suggesting that these serum markers could potentially be useful biomarkers of disease severity as well. This is corroborated by another study that also showed very strong correlations between immune markers and SCORAD. And we also have used serum proteomics as a tool to define different AD disease phenotypes across AD heterogeneity. For example, here, similarly to what we did with tape strips, we investigated the serum profile of AD patients, infants through adulthood, so across childhood maturation, and found that early AD, so infants, already had substantial systemic inflammation, particularly in the Th17 axis, as well as innate immunity. We've also looked at the blood of aging adults with AD, and we found that adults who were older with AD, so age 60 and up, were associated with a much higher systemic inflammation compared to both younger AD patients as well as age-matched controls, suggesting that aging and the AD disease state may both be synergistically contributing to a state of systemic inflammation and cardiovascular inflammation. We looked at different disease severity levels, where we found that mild AD seems to be a distinct entity compared to moderate to severe AD, completely lacking any systemic inflammation. And finally, we also investigated different ethnic groups and racial groups and the molecular endotypes that they represent. So here, for example, we compared an African cohort from Tanzania to a Swiss cohort from a Swiss-European cohort, where they both had distinct immune profiles, where some of the things we saw, for example, were that Th1 and Th17 signaling was more pronounced in the Tanzanian cohort compared to the Swiss cohort. We also looked at an Asian cohort of AD patients and measured several serum cytokines there, finding that serum levels of various Th2, Th1 markers were all higher in the Asian cohort compared to a European cohort. And then we also did serum studies before and after various treatments as well. In this case, after 12 weeks of cyclosporin therapy, various immune markers improved, among which CCL13 and CXCL10, their decreases correlated most with improvement in SCORAD score. And then with dupilumab, we also saw serum reductions in total IgE levels, CCL17, CCL18, and periostin levels. So to conclude and summarize, I think we've really come a long way in our understanding of AD. And really to further that knowledge, though, larger scale studies that recruit larger sample sizes across the entire heterogeneity of AD is needed. And in order to do that, that really necessitates minimally invasive sampling. Using tape strips and blood, we've been able to identify novel biomarkers of disease severity, of therapeutic response. We've been able to do longitudinal studies that evaluate for predictive biomarkers. We've been able to, again, study these distinct AD endotypes, which really helped us understand AD as a heterogeneous disease. But at this point, I think looking towards the future, refinement and application of these minimally invasive sampling techniques, particularly tape stripping, in both research and eventually in the clinic, will be really key to driving therapeutic innovation in AD towards a goal of more personalized medicine and to offer more individualized treatment options that are specific to a patient's unique features. And with that, I would just like to say thank you to my wonderful team at Sinai, as well as our collaborators. I'm happy to take any questions.
It had a very nice talk. So the quantified layer of the skin are mostly dead keratinocytes. So what's the source of RNA from the tip strips?
Sorry, I'm trying to catch the last part you said. So are you saying besides keratinocytes, what's the source of the cytokines? Or?
So what's the source of the extracted RNA from the skin tip strips?
What's the source of the RNA?
Yeah.
So we are getting keratinocytes, which are also themselves expressing a lot of these cytokines, and we also do get keratinocyte markers, of course. But I do think that there is diffusion of markers or cytokines, chemokines, other signaling molecules from lower in the epidermis that are diffusing upwards and being captured in the stratum corneum as well. So maybe even though there are not physical T cells that are like producing these markers specifically in the stratum corneum, I think that a lot of these mediators that are being produced deeper down are moving there, are diffusing into the stratum corneum that's being captured.
The second question is how challenging it is like to extract RNA from these tip strips.
I'm sorry, the acoustics are a little difficult. Can you repeat
so
that one more time?
how challenging it is like to extract RNAs from these tip strips?
Yeah, so we've really refined and optimized this method, and it does require a lot of thought because we have to figure out like what types of adhesives to use, like where they minimize the irritation, but you're still able to get the RNA yield. We have to kind of figure out like, you know, how many tape strips, like what's the protocol, how to best do it. But I think through some trial and error, we've kind of optimized it to the point that we are reliably able to extract the minimum, honestly, like far beyond the minimum RNA and protein yield that we need for these analyses. I think on average for RNA, we usually get like 40 to 50 nanograms, so we really just need 5 nanograms for these studies. But so through some strategy and optimization, we've been able to pretty reliably get the yield we need.
Helen, thanks for such a great review of the work. Just wanted to make a comment because when the initial translational work was being done, looking at gene signature markers in populations, we were a little slippery in terms of defining our population. So it has gotten much better. So I just want to make a comment that in clinical studies, we're very aggressive at trying to define our population. And in the science aspect of things, we're pretty rigorous at relating what it is that we're doing in terms of our evaluations. But sometimes in between, it gets slippery. And even in very recent publications in our highest level science journals, we've had translational research that's discussing groups of patients, like atopic dermatitis patients, that are undefined. There's no definition of the population, what their severity is, what standard outcome measures are used, even in the intervention. So I thank you for the movement forward. For instance, when Emma had her first data on biomarkers, Emma Goodman, and made the comment that, you know, back to it's a systemic disease predominantly. It was like, I wonder if we take a child with like 1% body surface area with minimal disease, if we're going to see the same systemic markers. And you actually did the studies to show that we don't necessarily see that in the less severe population. So it's just a call for continued, and when the population data came out about all children, you know, have a certain biomarker associated with them, yet we're separating out, you know, Asian populations from Caucasian populations. You know, obviously there's a mix of children in terms of their background. So, but the later work's much more specific, and I just think everyone sitting here who's doing translational work, please be careful in terms of your definition of the population so that it's reproducible.
Thank you for that comment, and I totally agree that at this translational level, more work needs to be done to confirm data at the population level, and it should not be conflated.
Quick question, quick comment, in fact, Robert Bissonnette from Montreal, about the presence of RNA in the stratum corneum, just to follow up on the first question of this after your talk. We collaborated together, as you know, in the presentation you made comparing psoriasis and atopic dermatitis, skin biopsies, and tape strips. And just want to comment that when we do tape strips on atopic dermatitis skin, we do get a lot of stratum corneum, but with some areas within an area of one single tape strip, we are sometimes within the epidermis and sometimes even lower. So in this study, for example, it was not infrequent that after 10 tape strips, we would see a very tiny drop of blood, suggesting that we are in the dermis. So I think we are getting cells that are significantly deeper than stratum corneum. It is heterogeneous, but we're deeper than stratum corneum.
Yeah, there's likely some heterogeneity there, and there's probably some differences in the tape stripping technique as well. I think I think when we looked at histology for our method, typically the most we got up to was the upper granular layer, but again, there's going to be variations depending on the exact tape stripping technique.
I would like to know your opinion on the use of serum Olink proteomics on the effect of Dupixent in AD, because some endotypes, Th2 high, were identified, but those do not predict the activity of Dupixent in the clinic. So how do we manage the information on proteomics with the activity of target biologicals in the clinic?
I'm sorry, I just want to make sure I understood the question completely. The question was on the utility of serum proteomics in evaluating dupilumab response?
Yeah, no, we know there are endotypes based on serum Olink proteomics that tell us there are Th2 high and Th2 low, okay? So then those patients that were Th2 high do not respond better to Dupixent in the clinic. So what's the relevance of this data on proteomics when they do not predict?
Yeah, I think overall, if we took everything on our samples on average, we do see in serum proteomics that there is a significant decline in these inflammatory and kind of AD disease markers before and after dupilumab. However, like you said, not everyone responds to dupilumab. And I do think that that's where molecular profiling, in the serum and in the skin, is really useful, where perhaps someone who's Th2 high, but has broader immune activation of other axes may not necessarily respond just to dupilumab therapy, but they may need a different type of molecular inhibitor, or something broader like a JAK inhibitor. So that's kind of how I unite some of the molecular data with the clinical data, and how they respond to one treatment but not the other. And I think this type of translational work gives us insights on who might respond to dupilumab or another treatment compared to who would not be responsive.
Hi, my question is, now I'm doing this transcriptomics on this inflammatory alopecia entity, and now I found some differentially expressed genes, and most of them are inflammatory cytokines, just like what you presented earlier. My question is, sometimes this inflammatory response is caused by maybe the sun exposure, or maybe the microbes on your skin. It would be very hard for you to differentiate whether this inflammatory response originates from the nature of this disease, or the exposure to the environmental factors. So I think I don't know whether you guys have any instructions on how am I gonna find the key molecules that belongs to the nature of this disease instead of the environmental factors. Yeah, I think that would be very helpful for my future study. Thank you.
Yeah, no, thank you for that question. And it's something that we grapple with, of course, in our analyses and our data too. First of all, kudos that you're able to get tape stripping in alopecia patients we find that it's very difficult for alopecia patients to be willing to tape strip their hair, their scalp, so kudos to that. But yeah, that is something that we have consideration for, like what part is environmental, what are contaminants. And then there's just several things that give us some reassurance regarding the fact that what we are noting in tape strips is actually disease-related compared to artifactual or environmental influences. As I mentioned, like we've done analyses where we took the first tape, we discarded the first tape. The first tape is, I think, where like that environmental influence would be most prominent. Looking at different tape strip layers, we really didn't see any differences. That was corroborated by a study that I referenced as well. The other thing for me is when we are doing our analyses, we are comparing a disease state to a healthy control. So if there were environmental influences, if we have a large enough sample size, the effects of that environmental influences on average should be similar to a disease state versus a healthy controls. And when we're looking at fold changes, then those fold changes, I think, are more likely to be related to the disease state that you're studying. And the other thing with that is we've done this in different diseases like AD, psoriasis, HS. There's probably a couple more now at this point. And they are all known to have different disease signatures that has been validated in biopsy studies as well. So the fact that we're able to see distinct disease signatures really tells us that it's not really artifact, it's actually the disease process, because if it were from artifact or environment, we would probably be seeing like something very similar rather than the unique features of each disease.
So it might be helpful if I can compare it with like other kind of alopecia entities to rule out all these artifacts or environmental factors.
Yeah, and I think correlating with biopsy data too, because a lot of these alopecias have been defined, like their endotype or their molecular phenotype has been defined in biopsy studies. So if tape strip data correlates really well with biopsies, that's probably very reassuring too.
Thank you.
Yeah.
I have just a quick question on your tape stripping data in very young infants looking at TARC. Is this assay robust enough to make a therapeutic decision?
Yeah, I think yes. Our data was pretty statistically significant, and I think the results of our transcriptomic analyses, even in infants, can provide insights into what would be a reasonable therapeutic option for them. I'm told that that has to be the last question, but thank you everyone for listening and for your thoughtful questions.